So then welcome to the issue about questions, and I fast forward. Science is question, but about science, okay? So the other questions we table for the time being. Again, we can see protein 3D structure, 1D structure is simply strings. 1D, 2D is this, contains the same information. When we look the superposed structures, we see some regions where they're similar. You see the helices evolving, we see regions where they're different. Typically, there is more movement in the ones where they differ. You know dynamic programming, you know the brute force way to compare sequences. Smith-Waterman does a local alignment with a gap open and gap extension penalty, so making gaps opening difficult but keeping long gaps relatively easy. Find this optimal superposition given an objective function such as this one. Identity is not really the best criterion. There's a BLOSUM matrix that looks at similarity of residues that say that matching the diagonal is important but not always the most important. Here the diagonal matches only 2, while there are many off-diagonal elements. Here's one highlighted that is higher than 2. Dynamic programming is fast when you align pairs. It's not fast enough when you could do 1 against 85 million. It's certainly not enough if you do 85 against 85 million. Then you need to speed up. You need to speed up by hashing. You know BLAST. Essentially you match a word. That's the hashing, that's fast. The problem, the challenge here is the statistics, getting the statistics right. So getting right the difference between the hit score and the background distribution, and simulating the background distribution from in an appropriate way is the trick of BLAST. I showed you that when we intrude into the twilight zone, we have an explosion of the true positives, pairs of proteins that have the same three-dimensional structure. But we have even a stronger explosion of the false true negatives, so proteins that have in fact different structures. So the red shoots up as an exponential scale, this number of protein pairs here. The blue shoots up by more than an order of magnitude. The red shoots up by more than 5 orders of magnitude, so much more. So already sort of on this side of the twilight zone of the H-val, you see at some point here in the H-val of 3, you reach sort of 50% accuracy. That means that the pairwise comparison alone is not enough. You need to sort of get into multiple alignments. Problem with multiple alignments, dynamic programming no longer works. Even if you could do up to 6 with 60 terabytes of mainframe memory, if that were possible, 6 is nowhere near where we want to go. We want to go 1,000. Cannot be done, can only be done by hacks. One hack was proposed by, this hack here is proposed by Rost, and I'm going to show you that one. So we want to align 4 sequences. What we can do is pairwise. We do an all against all pairwise. We get the percentage sequence identity written here. A to B is 90, A to C is 80, A to D is 70. You sort, you take the highest pair. Say you begin with A and B. Okay? So first you align A and B. And in aligning A and B, you create essentially a consensus sequence. The consensus sequence, wherever they are identical, you take the letter that they both share. Where they are not identical, now you have a choice. You can take the letter from the first sequence, or the letter from the second, or you can take the most informative. Here in this example, I take the one from the first. Okay? So this gives you a sequence. Right? Now, in the next step, you may choose the next most similar pair. Here, proteins. You align these two, same way, create a consensus sequence, and then obviously the next step is to align a pairwise alignment of the 2 consensus. Right? This is one way of doing it. Do you see another way of doing it? Just the same hack?
Iteratively do a consensus sequence.
So you can do step 1, you do this, and then you take the consensus sequence and you align it, for instance, against the third one here, right? So you create from the first pair, you create a consensus. This consensus is used to align against the third. And the next step, the next step is missing here. In the next step, you simply would take this consensus sequence and align against the fourth. That allows you to go a little bit deeper into the twilight zone. By the way, the third step, finally, I don't understand why I put the slide in between, comes here. Now, this is a simple hack. It is very simple and it's great. The problem is that there are alternative ways of doing it. So there's no rigorous mathematical treatment for the story. It's mapping problem onto something that you can solve. It's very easy. It's pairs of sequences. But again, how do you exactly define the consensus sequence is not well defined. That's the first problem. The second problem is you create artificial sequences. So everything so far here, these are sequences, actual sequences. The consensus sequence is one that may have very different features. So aligning this object with different features may no longer be the right thing under the same substitution matrix of a BLOSUM, because this is not existing in nature, the consensus sequence, right? So you open a little bit of a can, and that little bit of a can gets into perceiving what you do in a more mathematical way, and that gets us to something referred to as profiles, position-specific scoring matrices, or motifs. And this slide comes from Teresa. Thanks for that, Teresa. I've been showing this slide in many lectures. This first one where you're actually sitting in there. So the idea, it nicely illustrates the idea behind the position-specific scoring matrices. This is for the transcription factor binding side. Transcription factor binds at that point. You see this is nucleotide sequence. The height of the letter is proportional to how often that is used at that particular position. So 1, 2, 3, 4, 5. Upstream of the binding site, downstream of the binding site, negative numbers. You see there is a position in -7 where essentially largely is only C, while at -6 is only T. At +6 you have only C. So you can see that essentially you can use the observed count to come up to a probability, right? You can carve this into regular expression. So at every single position you can say L or I, you can have different motifs. Now the motif is a way of doing it. The trouble with the motif is not easy to put that into an alignment method. We know how to create regular expressions, we know how to use regular expressions, but it's not that easy to align regular expressions. So that's in some sense why regular expressions they are interesting, but they don't have a big future unless we get to the story that is more rigorous in terms of mathematical treatment, and that is essentially the profile that can also be done for a short region, and then it's a motif. So we begin with these 5 sequences here labeled 1, 2, 3, 4, 5, 6 positions, so 6 nucleotides in this particular case, and then we compile for initial alignment. We compile the absolute frequency at every single position, transfer that into a relative frequency, transfer that into a probability, compile log odds matrix. The log odds matrix will have the problem of infinity here, so we will correct that by pseudo counts. We will simply introduce counts that don't exist, small numbers, simply to not turn that into infinity, because then we cannot do math, or trivial. And that will ultimately give us a matrix that will replace this one here, the BLOSUM matrix, by a specific matrix. So now the protein is shown in running down here. Position number 1, first residue. Position 49 is the 49th residue. 4 protein sequences are aligned here. The first, second, third, fourth protein, first residue. Say position 11, you see that the first protein is an M, the other one an L, and then 2 times I. This is the count. We put the count again into log odds matrix, and the log odds matrix is shown on the right hand side here. This is not quite the log odds matrix, so this figure, this is a slide from me. So this figure is wrong. Wrong in the sense that the values here are not explained by this alignment. So the values are created on an alignment that is currently not shown. So it's a larger alignment. So that's a mistake. I have to somehow change this slide. And you see that here, for instance, at position 7, SSQE, and then the consensus is a D. And if all you ever see in that position is SSQE, consensus D does not make a lot of sense.
I have a question about that. So the matrix, we have a matrix like this for every position, right?
So that is correct. So, well, the matrix in this particular case, you have a vector for every position. So you have a vector of 20 values for every single position. The matrix comes because you go from residue 1 to residue L for a protein of L. In this particular case, L is 49. So that makes the matrix, right? Yes, you have 20 values at every single position, at every single residue position.
No, well, let's say we had a fifth probe, and
yes.
For each position, I would have, let's say for each position we have 20 possible amino acids. Yes. That fifth probe.
So the fifth probe will be exactly aligned according to this matrix. So you will put, say the fifth probe reads C, then it's -2. Let's just read, the next probe would read, L or M would be, here, V, it should start with a V.
That's not what I mean. So what I mean is, like, for the first position in the fifth probe, I would have a matrix, not a row, I would have a matrix for all possible scores.
No, a row.
Well,
for the first residue or for the entire protein?
First residue.
No, for the first residue I have exactly this vector, 20 values.
Yeah, but I don't know if it's a value, right? It can be any of
the next
20.
The next probe is the next protein sequence, and the next protein sequence will have a particular letter there, 1 of 20.
Okay, that cuts out the row.
Yes.
Okay.
Oh, now I get to the point. So, yeah, well, actually, so you could imagine that you create a sort of an artificial matrix now.
Yeah, precisely.
For
every position, I would have like...
which would be at every position the fitness of any residue at that position. Yes. But typically the problem that we are trying to solve is you come with a particular protein sequence. Which breaks this matrix. For those of you who did not understand Anton's question, that is on a different level. Let's ignore it for the time being. So, again, when you come in with a new sequence, you face a matrix. Because the new sequence has 1 to L residues, right? And for 1 to L residues, you will look up a position 1, 2, 3, if you line 1, 2, 3, what is the score of that, right? Is this clear? There's a lot of question marks in the room.
So for every position you have a matrix like this?
Yeah, Anton, I did not... that was a very...
Anton's question exactly, so, right?
That is Anton's question, but let's just assume... I hate to do this to you now, but let's assume this was not leading anywhere positive.
All right.
Anton had a very interesting positive point, but it's too complicated to discuss this point now. And since it is too complicated, I redefine his point as boring and wrong. Okay? Yeah. So forget it. Well, yes, ouch. But so since Anton has been argued under the table, what we have here is every single position we have 20 value. Okay? Anton's point is one that is valid if we had a different problem to solve. For this particular problem, at every single position you have 20 values, a vector. Okay? In a universe where you try to theoretically solve all the possible protein alignments, we're not talking about a vector. There we talk about a matrix, in a matrix, so to speak. That's more complicated. That was Anton's point, and he's right on that. But for bringing in a new protein, essentially at every single point here, your new protein may have the letter F at this point. And for the letter F, you can simply look up. Position 14, F is -5, or something like that, right? So it's discouraged.
And does the consensus get updated with every new?
Yes. So the consensus motif, and this is in fact exactly somehow what went wrong here in this slide. So for position 7, the consensus is not for those 4. It has been updated because the alignment has been longer.
So the answer to do we have a matrix for every position is no, because we have the vector for each position, and the matrix is just a combination of those vectors.
Yes, right? Anton agrees. But not answering. So ultimately profiles profit from the relation of families, so we have to build up a family first, and from that family evolves some local relation, which again you may call motif. So that brings us to the PSI-BLAST idea. The PSI-BLAST idea is to sort of begin with a generic scoring matrix and evolve into specific, position-specific matrix. PSI-BLAST is based on something that F Altschul has started, who's a senior investigator at the NCBI. He did his education in math and statistics and biology, master in Harvard, PhD in Harvard. In 2014, just barely 70 publications, but over 50,000 citations. By now it's over 120,000. 2 papers of his over 120,000. So the H-index is not that stellar, but by all means, this is one of the most cited, most visible person in the world of molecular biology or the world of bioinformatics, certainly. Here's another main author on the first paper, J Lipman, director of the NCBI, master at Brown, an MD at Buffalo University. Again, the number of publications is not that high, or was not that high in 2012, H-index higher, but again, on the BLAST papers, one of the outstanding people to coin many aspects of computational biology and bioinformatics. PSI-BLAST was published as a partial dynamic programming algorithm in '97. Here's the original paper. Again, this is now higher, over 60,000 for this. Other people in the group were Webb Miller, who went on and sort of sequenced the first mammoth. That's one of the things Webb Miller did. Tom Madden and David Lipman is seen in the back here, and Alejandro Schäfer. The sketch of the solution is very simple. You begin with a fast hashing. So the fast hashing, essentially you find the words. Then you have the issue of how to extend the words. There's the original extension that uses sort of a neighborhood extension, neighborhood substitution extension in the way we did before. I'm not going to go into the detail here. To the improved version over that now is the dynamic programming. So in between the matches you do dynamic programming. Why is this fast? Dynamic programming is order of the length square, and is a slow algorithm. Why is it fast to extend here with the dynamic programming? Why can we afford to do that? So BLAST is about fast, and now in order to sort of extend between the fast or the sort of simple word matches and slightly extend it over the word matches, I do dynamic programming. How can that be fast? Why not dynamic programming from the start? Is it trivial? You guys have to help me out. Is that the problem where everybody knows the answer and you just feel bored and believe I trick you? Or no, it's not trivial. Okay. Okay, the story is again that the dynamic programming is essentially in the order of the length of the protein squared, right? Say I found only one word in the middle. Okay? That one word in the middle cuts the protein into half. 1/2 times 1/2, so L squared is 1/4. So essentially, by that you reduce the time by a factor of 4. So the first hit you find somewhere in the middle, you immediately reduce this time that the dynamic programming would need from that. Given this seed, this anchor, this word anchor, is 1/4 of the original length. I mean, 1/4 of the original time for the full length, right? Half the length time, half the length is 1/4. And in this particular case here, you see we have 5 hits, and this is in fact not unusual. So the more hits you have in the initial finding, the faster you break it down. To the point, this is, the hashing is linear, and you add just a little bit more for very short stretches. Simply by breaking down the problem into finding a dynamic programming between relatively short fragments, you speed it up tremendously because it's L squared. Okay? Clear? Then the major challenge, I said, is about getting the statistics right, so the difference between the hit and the background distribution. Now, you do all of this, you build up the profile by saying, well, as long as the E-value that comes out of it, as long as the hit is far away from the background, I add them to the profile. This is what you collect by collecting the pairs, building up the profile. Then you have all the pairs, so you line the sequence against the database, and at the end of it, you have all the sequences that that are such that the background distribution is far away from your score, high E-value. You build the profile, and then that's the PS in the PsyBlast, and what's the I standing for? To do it again. So you iterate over it. Do it again and again and again. And ultimately come up with a refined profile. Let me illustrate what you do in running a PsyBlast. So say the green one here were your protein family. And again, for most of this lecture, family is defined as everybody who has a similar 3D structure. Okay? And a sequence, some sequence relation. So this is the unknown protein, or the query protein here you start with. So for what I showed you for the HSSP value, what you essentially do is you define a circle around this U. You can find, through pairwise comparisons, you can find everything within a certain safe similarity, shown in blue, of U. Now assume that in my particular database, for my particular search here, I find those 5 proteins. The 5 white ones, right? And you see these 5, they are not distributed equally around the center U. And why should they? It's actually very unlikely that they're equally distributed, right? So they are more on this side. Okay? Now this means that when you build now your profile from these, then the profile in fact does not create a circle. The profile creates an object, egg-shaped here, and that is sort of biased by where did you find these 5. Where do your sequences in the first round, in the safe zone, the pairwise alignment first round, where do they fall? And in this particular case here, they fall a little bit to the right. Since it's a profile-based search, you can make the surface, the area of this egg here is bigger than the blue circle. You can search further away, okay? Now, one particular reality though is if there had been a protein here, you'd have lost it. So it would have come up in the first iteration of PsyBlast, but in the final profile in the end, it's no longer in there. So when you go down PsyBlast, and this may originally was a feature, maybe this has been fixed, but you may have lost some things that were very, very strong hits in the previous iteration. Okay, but now you take this and look on, and by looking on, oh, here's something else that we don't find in PsyBlast that this one points out. So we could have now taken all of these 5 that were in the safe zone in the beginning, safe zone of hits, and triggered with each of those new blast pairwise hits. So for each of those, that would have been this light blue here, right? For each of these 5, you find a circle around it. Taking this, one of these 5, each of these 5 as a center for a new search. And every single time you do pairwise, you do find a circle around it, right? And in this particular example, again, you see that these disks, overlapping disks, by doing that, in the way I choose it here, are in fact the largest area than the egg. That in fact happens. So there are methods out there, customized blast searches that do exactly that. They take the first round findings, start new searches with those. The problem is this immediately or quickly gets into very CPU. I'm sorry, into very CPU greedy research, but sometimes it helps you. So this is something that you have to be aware of. The next thing to have to be aware of is assume that the egg at some higher iteration point would be so big that you include something from a different family. A mistake. Now assume that the part that comes from the green family at this point is 1,000. And from the wrong one you take 2. Does it matter? So you may argue it doesn't matter because 2 in 1,000 is a small number. But The problem is that this one here completely destroys the signature of the profile. Remember, I showed you in some examples, I showed you there's one column that is entirely conserved in a protein, right? And exactly those 4 conserved columns would no longer be conserved. Every one you had inlined until that point had the same residue or was fitting to a certain profile. And this is no longer relevant, because in this new one that you hit here, this does not have to be conserved, because that in fact is not a signature, because that thing has a different structure, right? So this is how very few proteins can completely pollute a profile in very, very quickly, yes?
I'm looking at the profile here, and there's a column with G's and then there's an N in the middle. So it does have some robustness.
It does have
it would survive those.
So again, there are 2 things to say. Is that the one you looked at?
Yeah, exactly. And the middle one, that's all G's, right? And then there's an N in SS81 yeast.
I'm sorry, I'm not entirely with you. The middle one is all G's here.
Exactly.
Yes. Yes, there's some robustness. That is true.
Okay.
But in this particular case, there is so there's no negative charge, for instance.
Okay.
There's no tryptophan, or there's no large residue. There's no triresin, no tryptophan. So we have an example. He's totally right. So this is not only glycine here. There's an alanine. There's an arginine. There's a lysine. I believe I saw. No, I didn't see it. Lysine, arginine. Well, I cannot also read. There's an NR and A. But okay, we have 4 amino acids of the 20 at that position.
Less than 2 out of 1,000.
Yes, but the point again is, if you now come with one that brings a new letter, this could matter a lot.
So if I bring a tryptophan in, that'll break my...
It could break your profile. Simply because it sort of breaks. Again, there are points here where, in fact, you have a bunch of residues that are allowed, and they come with a certain probability, and one would not make a difference. But it's conceivable that one makes a difference. A histidine triad, where you have to have 3 histidines at 3 particular positions, and suddenly you don't have a histidine there. Okay, it's a low probability, but it allows you to bring in things that are not histidines. And until that point, for the first 1,000, you always insisted on the histidine. So maybe this example maybe you have more of a point here than I on this example. Histidine triad is a point where I believe my point is more clearly made. Anyway, the point is that the pollution can happen because it violates the rules that have evolved before. Now there are different alignment methods available. Has anybody ever heard about Clustal, the Clustal series of methods? Can anybody say what they are compared to... I'm going to sort of have a quick run through a couple of things. So there's a generation of methods. Clustal X is the latest one. And ultimately, they use the phylogenetic tree to choose the pairs and do a correct pairwise alignment. So they're down to the rule of Glass-Doodle, and they do the... Should I close windows or?
No.
No. So to do dynamic programming on pair alignments, and for every pair they align, they choose the right matrix according to the similarity that the pair has. And I believe I'm going to keep it there due to time. And I'm not going to go into phylogeny. The story about phylogeny is essentially we are not able to really reproduce the past. Clustal Omega is the latest version. Clustal Omega has some issue with time for large families, but it clearly is one of the best methods out there. Has over 100,000 citations, and again the major person behind it is Des Higgins. The person who is sort of, Des Higgins is the one who understands the algorithm. Toby is the one who understands alignment. So Toby's an expert in manually, visually aligning proteins. And essentially the Clustal series is a combination of somebody who is the programmer or the intellectual mind, the algorithmic person who is Des, and the one who really understands the biology on this. And the third person, Julie, is coming into this. Julie Thompson, who really brought the front end, so the nice front end, the colorful way of showing it. I believe for Clustable, that was one of the main reasons why it has so many citations, because it's easy to show. Here's an older algorithm that also uses something similar, Maxon, and from Chrysander and Reinhard Schneider. That is the group behind the HSP value that I showed you, the length versus sequence identity. Essentially uses something similar as Clustable, but the important difference here, and that I want to point out, is the one between protein-centric alignment that PsyBlast and Maxom have, and the family-centric alignment that Clustable, and we get into the HMM and T-Coffee in a moment. The difference is, what I mean by protein-centric view is you begin with a sequence, and that sequence has no insertion and no deletion. That's your protein, and you try to find whatever is similar to it, sort of indicated here, right? When you find other sequences, they may have insertions, they may have deletions, but your start sequence, your query, is a real sequence, and you don't allow it to be changed. Insertion doesn't exist, right? While the family-centric methods, what they try to do is find the best alignment that sort of reflects the family. For those, no sequence here has any priority of any other. The first one is not special, right? Here, the first one is special, and by the fact that the first one is special, the one that is most similar to it is special. So with the first, with sort of labeling one sequence, actually everyone is sort of subdued to the first one being master. It's the protein-centric view and the family-centric view. And they both have different applications. It's just important to remember that, because whenever you do something for one particular protein, you want to predict its structure, you want to predict its function. This is not what you want. You're not predicting the function for a family. You're predicting the function for a protein. So you want an alignment method that somehow reflects the protein-centric view. Okay, so another way to do the math for the profile is the hidden Markov model, where essentially you have states. The state, you always know the state of the one state before you, and you have transition probabilities. You can transit to its own. And the slide here comes from Marco Punta. I have to change the slide, who is now at the Cancer Center in London. One example, I didn't change the slide. One example for which he shows this is you have Augsburg, a football team in Germany, playing, a few years ago, playing against many other teams, and essentially you draw a vector that Augsburg wins, Augsburg loses, Augsburg plays a draw. It's a probabilistic model. In this probabilistic model, you simply have the three states. The probabilistic model sums to 1. So one of the three states is what is going to happen in a game. And you're trying to sort of predict this outcome from some other value. For instance, whether Augsburg plays at home or somewhere else. Okay, so the H for home, the A for away. So essentially you have an input H or A, and you want to probabilistically predict whether they win or lose. And the simpler way to do that is simply, you have all the transitions written here, and you fill it with the data that you have observed. And you hope that from the observed data, by getting the transition probabilities for the entire model right, you can predict for the next game what will happen, right? And this is in fact the formalism. There are finite states, discrete alphabet of symbols, there's probability transition states, and an emission matrix. Let's get into types of HMMs. This essentially is an HMM that sort of models the transition from red, yellow to green, and from green to red again for the traffic light. This is not probabilistic though. This is completely deterministic. The sequence will always be exactly the same, right? This is not a probabilistic model. While this model here is one that is highly probabilistic, highly non-deterministic. So whether it's sun, so you observe one of these traits, and you want to compute the probability of tomorrow observing another state. Sitting in Munich, most likely, you observe sun today. You know already it's not going to be sun tomorrow. The weather changes every day these days, right? But other than that, it's a relatively simple probabilistic model that's very complicated to do in countries like this one. It's a first order Markov, meaning that you essentially only, the state from tomorrow is based on what we have today. That can be used for alignment, and alignment methods that use that go back to Anna Schroch and David Hausler, Kevin Karplus, so that was the Santa Cruz model, and Sean Eddy, the Hammer model, who took it to other levels through the PFAM database. The way to do that is essentially you have a start, then you have a match state, that means you align two residues, or you have a deletion state, or you have an insertion state. And for all of these transitions, you learn, for all of these states, you learn the transition. How do you learn the values of transition probabilities? Once you have that model, and you come with a new sequence, you simply compile the probability of going through the model, and you pick the way to go through it that has the highest probability. Okay? Essentially, you do them all, right? That's like, in some sense, the algorithm underneath is faster than dynamic programming. It finds is a fast shortcut to finding the optimal probability, but it actually threads it through the entire model. How do you get to this model? What do you need to start? You need essentially a bunch of sequences for which you know they are related, and you need to align them. And then you say, this is my, let's call it alpha alignment, that trains my model, and I use my model to go further out. This is like the iteration of PsyBlast. Okay? Just the same story.
But wouldn't that actually, so the way I see it, this model would actually just produce a consensus sequence.
No, the model no. The model produces a fit to the model, or the model does not produce a fit to the model. You have a model, and the Baum-Welch algorithm lets you find the highest probable path through that model for your sequence.
All right.
Which is not a consensus sequence. Yes, you can say, well, in some sense, I could say yes to what you say, because for a consensus sequence, you can compile essentially how much it fits. It's true, but essentially this is just a model which, so far, I called a PSSM, a position-specific matrix. This is exactly what it is, because every arrow here relates to an entry into this column there, and here we have the 1 to 49 that was on that slide showing downwards. So this is exactly the same story. Except for here, this is not 49, this is 4. Okay? In this particular protein. So we have many, many more match states. We can build up the model. And here's Sam. I'm not going to thank him for his slide, Kevin for his slides, because I'm going to go through the slides very quickly. That's simply sort of, I zoom over them. It shows you how you essentially, by adding information, just like in PsyBlast, change the profile step by step, build up the profile, get to a point, and then you do the iteration just like you do in PsyBlast. It's the same thing as the iteration in PsyBlast, except for the math is different. The underlying mathematical model here is a hidden Markov model, okay? And the hidden Markov model lends itself very much to the sequence comparisons. That's why it has been so successful in the field. That gets me to the next level here of tea coffee. Genetic algorithm. Anybody has an idea who has seen tea coffee or has seen genetic algorithm for alignment? Anybody knows any of that? No? So what can it do that the others cannot?
Well, the others assume independence, right?
So that's the major problem. So whatever we talked about, dynamic programming, PsyBlast, SAM, all of those essentially assume that what happens at this position here is independent of what happens at any other position.
No, actually, I'd like to rephrase it. The genetic algorithms don't assume
don't rephrase
anything.
it.
That's the point. They don't assume anything.
Anyway, so this is correct. This assumption is not needed by the genetic algorithm. But again, let's get back a step. Let's slow this down. Everything at this position is independent of what happens at any other position. Is that true?
it really depends on the sequence itself and the structure it has, but if you like have a really big or a charged residue at some point, then of course it will influence what residues can get into it close to this position.
I believe this was clearly saying no in one word. It's not true. Does anybody want to defend that it is true? So okay, let me repeat. You have heard about dynamic programming, you have heard about PSSM. I've already told you that the most quoted papers use dynamic programming or PSSM. You have heard about SEM and Hammer. Those are the methods that use PFAM. It's another big quoter. So most of the, I don't know what most means, 99% of all the alignment methods out there use this assumption. So you want to tell me they are wrong?
They're not wrong. They're a model.
So they start from an assumption that is wrong.
I think the problem is that in order to check back the dependence on any previous members of the alignment is pretty hard and how to evaluate it, and probably that's why they don't use it, because the way to say probably biologically there is dependence. But
so actually,
in order to evaluate it, it's pretty hard, so they assume dependence.
Gary, you make a point that that is very correct, and I very much disliked by me personally. So, but it's sort of let's call it a personal vendetta. I'd argue this is very easy to validate, that this assumption is wrong. Whatever it is, the fact is they all make the assumption, and the assumption is wrong. As Anton so happily says, this is a model, this is not reality. As Gary correctly points out, dropping this reality is not easy, but there's genetic algorithm. That does not make this independent assumption. That does not need to make it, because essentially it operates on segments. Example for a genetic algorithm here is this spacecraft antenna, the shape of which is optimized by genetic algorithm. You cannot quite see that it is, but it looks odd. It looks unexpected. So you start with initialization of a population, you compute the fitness, and then you get through a bunch of changes here. So you roulette parents, so you choose parents. You cross over, and upon crossover you take some from the mother, some from the father. You do some mutations, and then you compute the fitness. Again, there is a fitness function, but the big difference here, and in the other one in dynamic programming, in PSSM, there's an objective function, but the big difference here is the fitness function can be defined on a segment. While for all the others, fitness function, dynamic programming, they are defined on per residue base. One column I is independent of j for all j's, not equal I. And here this goes into a segment. Now the people behind it, T. Koffer is Cedric of Notre Dame, who was a student of Des Higgins. You may remember, and Jaap Herringer was behind it. So you may remember that Des, in fact, is behind Clustal. So the sort of way to go over Clustal comes out of the Clustal group. And by the way, this is Cedric's team and his workplace. So if you sort of would like to live in an environment like that, visit Cedric. It's in Barcelona. So this is really their building, and this is really they're really standing behind the group space, and that's really typical view from there. And you can actually go to that beach. It's just across the street. You begin with a library of local and global pairwise alignments. Essentially what that means is you take any alignment method that you have, PSSM, Clustal W, you let it produce a bunch of pairwise or multiple alignments for that particular family, and then you crank it into a device and you get a refined alignment, the genetic algorithm. The beauty of it, you can actually do even more. You can put structural constraints in it. So this has to be a helix, so there are certain residues that have to be next to each other, pairs of residues that are distant in space, you can put contact constraints in it, and some specialist information, that is a binding site, so they have to be close again in terms of contact. This should be in the same domain, or all of these kind of things you can easily put into the fitness criteria of the genetic algorithm, and out comes an improved multiple alignment. works like a charm. The only problem with it is it takes a little bit of time. CPU time, that is. So this is not the kind of thing you can do for running all human proteins against the database, but this is something that you can do for a particular family if you have enough CPU at your hands. So tcoffee can run many days for one particular protein family, for not so large protein family, but the alignments are really great. The more you know about a family, the better you will do with that method. So then there are other methods here that essentially, again, SS search is one that has dynamic programming, similar in terms of being protein-centric. Then there's an iterated SS search. This is a very, Bill Pearson here, SS search is sort of the modern FAST-A. There are a couple of other, Muscle is another well-known dynamic programming method. There are many, many alignment methods, and ultimately what distinguishes them is the speed, which is important, and the way they use statistics and the way they do the alignments. The most prominent, mostly used for speed is PsyBlast. For doing nice alignments is the Cluster series, and for essentially getting a nice family alignment, family-centric alignment, is the HMMs at this point. If you know more, if you have more time, you run tcoffee. That gets us into when we get into the daylight zone here, when we look at how I can infer similarity when two protein sequences are very similar, it's trivial. We get into the twilight zone, we use profile. So here, in the daylight zone, pairwise alignments are good enough. Here we need sequence profile alignments. And if we want to go further, what can we do there? So we begin by sequence, then we do a sequence profile, and then we do, profile alignments. So essentially we go one more step. So rather than taking one sequence and compare it against a profile, you compare two profiles. Okay? Now, or even two sets. So one database of profiles. So you take the sequence and compare it against a database of profiles. The problem is, again, the space of possible options explodes here. So let's look at the simplest thing. Now you're actually not. Now we have truly the Anton solution. Or we're comparing two vectors. We compare this vector against that vector. Again, not thinking in tcoffee, so we ignore again that position I and j. We pretend that I and j are completely independent, so we do it residue by residue. We take that particular residue column here, where I have a vector, and compare it against that particular column, where I have a vector. We compare two vectors. Now when we compare two vectors, you all know there are different ways in which we can define the distance, or the product of two vectors. They're we can put vector distance, we can look at the Euclidean distance, we could look at the Manhattan distance. So there are different ways in compiling distances. Unfortunately, this is crucial here. And for some families, one of these ways works better than the others. And that's not family independent. So meaning profile comparisons are highly sensitive to how informative the profile is, what family I do, and that means it's getting tricky to do. So again, the free parameter space explodes. Here's a cheap hack of doing profile, and originally that came from Dariusz Przybylski, who's now at Broad. So we sort of start, PsyBlast does profile sequence. Profile is in terms of CPU complicated. It's complicated in the optional space. So maybe what we can do is we sort of find a way in between these two extremes, profile sequence to profile. Maybe we sort of find a solution that is halfway and very fast. And his idea simply was take the profile and compile a consensus sequence from the profile. Okay? So, and the consensus sequence in this particular case is completely artificial. So you're not actually looking for any sequence that you want. You're just going, so in this particular case your protein has 15 residues, so protein residue 1 to 15. You look up in the position-specific profile, you look up whatever is the highest number at every single position. And that's the amino acid you're going to copy. And there's some solution for what you do if two numbers are equally high. So you're going to have the artificial sequence L, P, P, R, oops, there's the R, and so forth and so on, right?
How do you decide on the score for each?
Oh, so that's the position-specific matrix. So this is essentially what PsyBlast does, or what HMM do. So what you see here is the simple profile, okay? Now you take the profile, is clear?
No.
Right? So you take the profile and convert the profile into a single sequence. And from here it's clear, right? So now you build up your database of consensus sequences, and then you essentially run a PsyBlast against the database. Instead of against the database, you run the same algorithm, PsyBlast, now against the database of consensus sequences, where essentially every single one of those contains information about a profile. They're all not real sequences, they're all sort of condensed profiles, if you want, right? So you can do that at the same CPU time as running a single addition to PsyBlast, or a single pairwise blast, ultimately. So it's rapid, it's very, very fast, once you have the database. There's a lot of overhead to produce the database, but once you have the database, this is just as fast as a pairwise alignment. And what this one says is it improves as much over PsyBlast as PsyBlast does over BLAST. Let's just leave it there. The more modern way of doing that comes from Johannes Söding, who was at the LMU and now is in Göttingen. The method is called HS Blitz, bless you. May you be blessed. Originally it came also from Andrei Lupash, so the latest development really, so the original idea was developed in conjunction with Andrei Lupash, but now the later things are really only pushed by Johannes. You have a query sequence, you build a query HMM, you essentially do the same thing, you do HMM against HMM, and you can gain speed that you need by not using the typical sequence, but translating the sequence into some consensus sequence that is not only using 20 letters. Okay, that's the trick that they use to speed up. And let's just keep it there. HH Blitz, I believe, is one of the best things, and there's now Jack Hammer that sort of tries some of these ideas, but I believe those are amongst the best tools out there if you want to go to identify distant relationships. Clearly the profile alignments of this type enter into the midnight zone, and that gets me to the midnight zone, from the twilight zone to the midnight zone. So my question that I'm now going to ask is, when we look beyond the twilight zone, how much is actually there? What I'm going to do is I'm going to compare all protein structures. I'm actually not going to do all against all, because there's a redundancy reduction necessary. So I take a redundancy reduced subset, compare that against all. And now my question to you, what I'm going to plot is a distribution of pairs of proteins that are similar in 3D structure. And I'm asking, at what level of sequence identity do I find how many pairs? Okay? So I plot sequence identity from 100 here, 50, 0, and I want the distribution of all the pairs of proteins that have similar 3D structures. What do you believe?
I believe that 50 to 60% sequence identity is sufficient to have a similar 3D structure.
No, but I'm looking at the set of all the proteins that have similar 3D structures, yes. And I'm asking, where do I see most? So do most pairs of proteins that have similar 3D structures have 100% sequence identity, or 80? How do you expect the distribution to look?
Well, the mode should actually be at 100%.
The max would be at 100%, so you expect something like this?
Well, more something that is logarithmical.
So something, almost never a straight line. This is already a very good answer. Almost never true a straight line. How do I do your? Like this?
Well, maybe a little bit with the plateau being like.
Wait, most sequences are not similar, right? So how can the mode be at 100%?
Similar. They have similar structures.
That is the distribution of like pairs.
So what do I do now? Anton has another. So Anton says we shouldn't start with something like this here. We should sort of ramp up to something like this. Is that what Anton says?
Well, I would say that if we're talking about structure similarity, then most proteins are not similar. Therefore,
that is correct.
The mean or but this is a distribution, right?
But this is the distribution of all the ones that are similar.
The question.
So I'm not asking how does this distribution look for everything. I'm asking for all the pairs for which I do a 3D superposition, and from the 3D superposition I say they have similar structures.
So how many out of the proteins that are 100% sequence identical have identical 3D structure? And
be careful, we have a redundancy reduction. So half of PDB is reduced at 100% already. But they are actually not going to be visible here because I cut them out.
all right. Then I have to think about it again.
I'm not sure I get the question. So we take only the similar proteins.
Proteins that have, you take the entire PDB and you do a shape comparison. You take a sequence unique subset, which you create sequence unique by a sequence comparison, right? Once you have the sequence unique subset, and this is h-val 0, so this is essentially no pair has more than 20% sequence identity over long alignments in here. And then you take this and compare the 3D shape of these 10,000 against these 120,000. So the numbers I show you is when the numbers were smaller. But today you would do here 10,000 against 120,000 3D objects. And the 3D objects you would say either they have a similar 3D structure, less than 2 angstrom or something like that, or 2.5 angstrom, or they have a different structure, more than 5 angstrom. Everything in between I throw away because I cannot decide. And I'm plotting now only what is less than 2 angstrom. So I'm plotting only the pairs that have similar 3D structures. And I'm wondering, what do I expect in terms of their sequence similarity?
They don't have to be very similar.
No, they don't have to be similar. The question is, what will I see in the PDB?
I would expect somewhere below 50% to be the mean.
So we are moving, and now the peak is something like, no wait, below 50.
Yeah, somewhere, yeah.
Something like this.
Yeah, probably.
Anybody has an issue? I mean, anybody says no to that because of some reason or other. Okay, so actually by the way, this is the random. So random superposition, 20 amino acids, roughly 5%, right? So this is the observation. Okay, actually, this is the expectation. There's some issue with. I'm not sure why I put this one here. There's some issue here with the counting, the PDB. There was an old slide, so I expected this to be higher. Anyway, so something like that, right? And something like that is not true. It's absolutely not true. The real truth is this. And mind you, so you put the maximum somewhere under 50. Here, this is 25. Okay? The peak is at 10.
It's under 50.
This is under 50, but this is here on this plot. And it does not look like this at all. It looks like everything is here. Compared to this peak, there's nothing here.
So the sequence doesn't matter, is what this plot is showing.
And for the pairs, ultimately almost. So now we get into sort of interpreting that. So red is random. And I would argue that the difference between random and the structure is exactly how a sequence matters. If sequence wouldn't matter, all structures would be the same, right? That cannot be. Sequence must matter. But somehow the space or the amount of residues that you have to play to make sequence better is the difference between the two. So it's not actually the value of this distribution, it's the between random and this. For random sequences, sequence doesn't matter and structure doesn't matter. they don't adopt the structure. The difference is that it's sort of all the information you have that is specific to these structures. 3 percentage points, 5 percentage points on average. This is remarkably small. And ultimately, then you have to remember somehow that many of those in between here are also binding site constraints. So it may not even be the structure that I see. So it's just the left. So ultimately what it means, I call them anchor residues here, is it seems as if the time we had in evolution was long enough to really get to a point where whether you have convergent, and this is a misnomer here, convergent to the same structure is what I mean here. It's not convergent in the sort of classical evolutionary sense, but between convergent and evolution and divergent evolution, you can distinguish if you are somewhere high, it's more likely that they came from here. But if you're that close to random, essentially you reach the point that most likely you can reach from both sides equally. Which means you somehow reached an equilibrium point. And this also means that most likely convergent to the same structure is a very frequent event. Now, it means that really to where we want to get, for most pairs, we will never get there. This is too little, even for profile alignment. So to find the 3% in an alignment that are specific, this is extremely challenging. And then ultimately, all the advances that have been done, have been achieved, for instance, by Johannes Söding, to push the profile hidden Markov idea, are so successful because you have an explosion of positives you find, are so difficult because ultimately where you really want to reach, the problem is almost impossible to reach.
So,
yeah?
So we're left with 3D structures if you want to do this exhaustively.
If you have to get those, you essentially, yes. There is one idea here to essentially look at secondary structure and use secondary structure for the alignment. And let that help you to go a little bit further. And what these slides say is that, and I'm sort of glossing over it, it works to some extent. The point that I want. So essentially, what you do now. Let me fast forward this. You take for every single position you predict whether it's a helix or strand, or other, you predict whether it's exposed or not. This gives you 3 times 2 states, exposed or not, helix, strand, other, 3 times 2 is 6 states. You have 20 amino acids, 20 times 6 is 120. Essentially you have now a letter in an alphabet of 120 states. And for these 120 states you can do a simple alignment. Simple dynamic programming alignment. That's sort of the idea here. And that works very well. So here's a very surprising reality where we look percentage sequence identity and then sort of the identity of pairs for properties that have a similar structure. So if the curves, the blue is higher than the red, is higher than the purple. So the blue is the highest. And the blue is surprisingly predicting 2 strings of predicted secondary structure. Meaning, while this one here we have observed versus observed, red, and down here we have predicted versus observed. So if you do want to find out whether proteins A and B have a similar structure, and you want to use secondary structure prediction for that, then you best do by throwing their observed secondary structures away. And by predicting secondary structure for both. It's good news because you don't need a structure. It's very surprising. Anyway, this method clearly improves very much over PsyBlast. You can go into the midnight zone with that to some extent.